Good morning. It's May 15th, and this is your daily brief in science. Here's everything you need to know. A newly identified long-necked sauropod from northeastern Thailand, Nagatitan chayafumensis, stands as the largest dinosaur discovered in Southeast Asia to date. The name honors Naga, and researchers suggest it could be Southeast Asia's last true titan, given shifting geography that may limit future sauropod presence. Classified in Euhelopodidae within Somphospondyli, Nagatitan is dated to the early Cretaceous, about 100 to 120 million years ago, in an arid to semi-arid riverine landscape. The study notes that sauropod gigantism evolved independently across regions, driven by climate, habitat structure, and herbivore biology. Independent experts not directly involved in the project, like Matthew Wadle, call the find a robust dataset that advances understanding of giant dinosaurs and regional paleoenvironments. The research was led by Thidivut Sethapaneksakul of University College London, with Paul Upchurch as co-author, and published in Scientific Reports on May 14, 2026. Fossils were first discovered in 2016, with excavations from 2016 to 2019, and a restart in 2024 after renewed funding. Experts highlight the find's significance for understanding sauropod size evolution and earlier growth trends toward later giant forms. Fully grown Nagatitan likely faced little predation, with threats mainly to old, sick, or juvenile individuals, suggesting rapid growth as a defensive strategy. The discovery comes amid rising CO2 and higher global temperatures, factors scientists link to sauropod biology, heat management, and vegetation. The ecosystem was subtropical, featuring forests, savannas, and shrublands that supported diverse dinosaurs, pterosaurs, crocodiles, freshwater sharks, and a large Carcharodontosaurus-related predator. Researchers estimate Nagatitan was a bulk browser with a long neck and tail, a small head, and 4 columnar legs, feeding on high vegetation such as conifers and possibly seed ferns. 2 gorillas are being traded between Boston and Pittsburgh as part of the Gorilla Species Survival Plan, with 7-year-old Frankie moving to Boston to join a bachelor group and 33-year-old little Joe relocating to Pittsburgh to join the family troupe. The broader SSP aims to balance social well-being, training, and breeding opportunities to sustain long-term health and genetic diversity for captive gorilla populations. Boston's Franklin Park Zoo and the Pittsburgh Zoo and Aquarium are exchanging individuals to maintain genetically diverse populations in human care. The piece uses a baseball analogy for the swap, including related MLB context to add entertainment value. Social media has framed the swap as a veteran for prospect move, reflecting fan engagement with the unique trade. Public online reaction highlights both the novelty and controversy of exchanging gorillas between zoos. The narrative emphasizes power dynamics and market-like negotiations in a parity sports trade frame rather than a traditional transaction. Sources cited include the Pittsburgh Tribune for enclosure plans and Fox News for related coverage, noting the sensational tone. Little Joe's background includes a high-profile escape in 2003 that injured a child, adding complexity to his history. The piece incorporates fictional elements and stylized insider figures to fuel humor and world-building in a sport news voice. The trade is framed with baseball language linking moves to teams like the Red Sox and Pirates for a humorous parallel to MLB strategy. Zoo New England's CEO described the decision as thoughtful and not taken lightly. A new meta-analysis of nearly 650,000 pregnancies exposed to antidepressants and about 25 million unexposed pregnancies finds no clear evidence that prenatal antidepressant use increases the risk of neurodevelopmental disorders like autism or ADHD, once maternal mental health, family history, and other confounders are accounted for. Initial signals of higher diagnoses fade after adjusting for these factors, pointing to shared familial or maternal factors rather than a causal effect from the medication. Some elevated risk signals show up with paternal antidepressant use during pregnancy or maternal use before pregnancy, reinforcing that the exposure during pregnancy itself is unlikely the driver. The lead author urges clinicians and patients to consider continuing treatment for moderate to severe depression during pregnancy to prevent relapse, while weighing individual risks and benefits. Experts not involved in the study emphasize the difference between correlation and causation, Noting maternal mental health and family factors can explain observed associations. Limitations include gaps in socioeconomic data, lifestyle information, birth weight, and trimester-specific dosing, along with residual bias from maternal depression severity. Strengths include adjusting for maternal mental health, but there was substantial heterogeneity across studies and missing data on key perinatal factors. Additional limitations call for richer, longitudinal datasets with broader socioeconomic and perinatal variables in future research. Sibling-matched analyses reduced observed associations, suggesting shared familial factors contribute to the findings. The study's conclusions support reassessing public concern about antidepressant use in pregnancy and basing medical decisions on comprehensive, confounder-adjusted evidence. No significant links were found for intellectual disabilities, motor disorders, or speech and language disorders. For mild depression during pregnancy, non-pharmacological approaches like psychotherapy may be suitable, but abrupt discontinuation of antidepressants solely over neurodevelopmental risk is discouraged due to risks of untreated maternal depression. A new method called Sempor-Cupine combines chemical labeling of non-paired RNA bases with direct RNA sequencing through nanopores to analyze RNA structures at the level of single molecules. Optimized chemical markers act as signposts for exposed RNA regions likely to influence folding, while nanopore sequencing reads the entire molecule to capture detailed structural information. This enables full-length single-molecule analysis of RNA structures and their role in gene regulation, health, and disease. The work is led by Dr. Wan Yue of Astar GIS, with co-led Dr. Niranjan Nagarajan, and was published in Nature Methods. Researchers say the method signals a potential paradigm shift in transcriptomics and RNA-targeted therapeutics, thanks to nanopore-based analytics. The approach provides deep insights into how RNA structure governs cellular outcomes and disease misregulation, with implications for understanding viral RNA function, including SARS-CoV-2. Applications may include improved diagnostics, drug discovery, and precision medicine through a clearer view of RNA folding. The study demonstrates a direct link between RNA structure and translation efficiency and RNA degradation rates, highlighting how structure can influence gene expression. By revealing how RNA structure governs pathogen gene regulation, the method points to RNA-based targets for therapeutics and antiviral strategies. Astar GIS plans to integrate Sempor-Cupine into its research infrastructure to accelerate precision medicine and diagnostics by decoding RNA folding at unprecedented scale. Sticky gel-like condensates formed by RNA-binding proteins trap viral RNA at replication sites in plant cells, blocking viral replication. Researchers from the CSIR Center for Cellular and Molecular Biology used nuclear magnetic resonance, fluorescence microscopy, and molecular dynamics to map how these condensates assemble and interfere with viral RNA. The study reframes RNA-protein interactions as dynamic, phase-separated droplets inside cells rather than fixed, membrane-bound processes. Led by Mander V. Deshmukh and published in the Journal of the American Chemical Society, the work shows charged surfaces on proteins create sticky patches that drive clustering into networks, forming a biomolecular condensate around viral RNA. This process acts like a molecular glue, hindering replication and spreading within plant cells. The findings point to potential agricultural applications, including crops with stronger innate immunity to viral diseases, aiming to reduce global crop losses. Beyond plants, the research hints at broader implications for human health, potentially informing therapies for neurodegenerative diseases and certain cancers, and guiding future drug development. The work integrates NMR data, imaging, and simulations to support the model of droplet-like compartments at viral replication sites. Overall, the study suggests a new framework for RNA-protein interactions and opens avenues for both crop protection and medical research against protein aggregation-related diseases. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.